Welcome to Metallomix, a pioneering force in the pharmaceutical industry dedicated to advancing breakthrough therapies that target inflammation and oxidative stress. Our mission is to become a world leader in Metallomix, focusing on developing inherently safe and effective disease-modifying drugs for complex diseases. We are committed to transforming the treatment landscape by addressing the root causes of these conditions rather than merely alleviating symptoms. Join us as we explore the potential of Metallomix to revolutionize healthcare and improve patient outcomes. While organic elements make up the majority of an organism's mass, life would be impossible without the metallome, the essential inorganic components. These elements play a crucial role in various biological functions, including amino acid and protein synthesis, DNA integrity and repair, charge balance, and energy storage. The metallome is integral to maintaining cellular signaling, stem cell function, and biomineralization. At Metallomix, we recognize the importance of the metallome and are dedicated to harnessing its potential to improve health outcomes. At Metallomix, we are connecting the metallome with other biological omes, such as the genome, proteome, and transcriptome. This integration is essential for understanding the complexities of neurological, age-related, and metabolic diseases. By modulating cellular processes through Metallomix, we can influence a range of biological activities, including metallenzyme function, DNA integrity, and mitochondrial performance. This holistic approach allows us to develop therapies that target specific organ systems while considering the broader biological context, ultimately leading to more effective treatments. The isotope selective modulation platform, or ISM, is at the core of our scientific approach. We start with isotopic signature discovery within a specific indication, followed by identifying metallome-specific biomarkers affecting a disease state, lead compound selection and optimization, drug candidate preclinical and clinical validation. This innovative platform enables us to target disease pathogenesis by shifting isotopic signatures in key biological molecules and address the underlying causes of diseases rather than just their symptoms. ISM platform represents a paradigm shift in drug development, offering a more comprehensive and effective approach to treating complex conditions. Our scientific stance at Metallomix is based on a whole-body approach to drug development. Traditional therapies often focus on a single target or pathway, which can limit their effectiveness and fail to address the entire disease cascade. Our approach aims to halt the disease process at multiple points, ensuring broader therapeutic impact. By avoiding negative effects on healthy biological functions, we can develop treatments that offer maximum benefit with minimal risk. This holistic strategy is the future of medicine, focusing on broader targets that address the root causes of disease rather than just symptoms. Metallomix therapeutic platform allows to achieve lasting health improvements, ensuring that treatments are more effective and sustainable, offering long-term benefits for patients. Our therapies are designed to target disease pathogenesis through isotope selective modulation. Disease is often associated with shifts in isotopic Metallomix signatures within key molecules, cells, and tissues that leads to the expression of proteins with disrupted structures, resulting in the disease aggravation by the induction of oxidative stress and inflammatory pathways. By modulating these signatures, we can restore normal biochemical pathways and reduce disease progression. This approach allows us to intervene at critical stages of the disease process, offering a novel method for addressing complex conditions and improving patient outcomes. Disease pathogenesis is influenced by unhealthy omics, characterized by exposure to environmental hazards and chronic stress. These factors increase the risk of DNA damage, protein misfolding, and cellular dysfunction. By correcting isotopic imbalances, we can reduce these risks and promote healthier biological systems. Our understanding of disease pathogenesis allows us to develop therapies that address the root causes of disease, offering a more effective and sustainable solution for patients. Our lead candidate, KLS1, represents a first-in-class drug with significant therapeutic potential. This patented small molecule conjugate is systemically bioavailable and capable of crossing the blood-brain barrier, It acts upstream of toxic protein formation, reducing inflammation and oxidative stress. KLS1 is intrinsically safe, demonstrates a linear pharmacokinetic profile, and has proven its efficacy in several proof-of-concept preclinical cell and animal models. KLS1 has shown its ability to restore normal biochemical functions in oncological, neurodegenerative, and metabolic preclinical models, making it a valuable addition to our advanced clinical pipeline. Zinc is a critical component in numerous biological processes, playing a key role in DNA recognition, gene expression, and apoptosis regulation. It is a vital part of many proteins, such as zinc finger proteins, P53, Parkin protein, and many others. Deficiencies in the zinc are linked to several devastating, life-threatening diseases, including cancer and various neurodegenerative disorders, highlighting the importance of zinc in maintaining health and the necessity to maintain a healthy zinc metallome. Our zinc-based therapies aim to address these deficiencies, offering a targeted approach to disease treatment. In the realm of human health, zinc emerges as a critical player, touching nearly every system in the body. From the brain to the bones, its influence is far-reaching and profound. In neurology, zinc deficiency is linked to neurodegeneration, neuronal lethargy, and various nerve conditions, while also playing a role in brain cancers and neuropsychiatric disorders. Moving to pulmonology, zinc's importance is evident in protecting against lung damage from viral and bacterial infections and potentially influencing lung cancer development. In metabolic health, zinc impacts insulin sensitivity and may help address obesity and fatty liver disease. Its role extends to nephrology, with implications for kidney diseases and cancers, and to cardiology, where it influences conditions like arrhythmia, atherosclerosis, cardiomyopathy, and coronary artery disease. Zinc's reach doesn't stop there. It's crucial in dermatology, urology, including fertility and testosterone production, oncology, and even in maintaining a healthy gut microbiota and immune system. From myocardial healing to bone and muscle health, zinc's importance in human biology underscores its potential as a target for innovative therapies addressing a wide spectrum of health conditions. Zinc-64, the lightest stable zinc isotope, exhibits unique physiological behaviors that are essential for maintaining metallome homeostasis. This isotopic fractionation leads to differential depletion of light and heavy zinc isotopes, affecting metallome balance. Our research shows that healthy cells have light isotope signatures, while diseased cells display heavier profiles. By understanding these dynamics, we can develop therapies that restore isotopic balance and improve health outcomes. Metallomix groundbreaking research has uncovered a fascinating aspect of zinc metabolism at the cellular level. The company has found that zinc isotopes are not treated equally by cells, with different kinetics governing their uptake and excretion. This phenomenon, known as isotopic fractionation, results in an uneven distribution of light and heavy zinc isotopes within cells and tissues. Over time, this process leads to a complex disruption of metallome homeostasis, which is the delicate balance of metal ions essential for proper cellular function. Metallomix studies have revealed that this metallome dyshomeostasis is closely linked to various disease states, manifesting as distinct isotopic signatures. Perhaps most intriguingly, our research has consistently shown that healthy cells and tissues tend to exhibit signatures characterized by lighter zinc isotopes, while diseased cells and tissues display a preponderance of heavier zinc isotopes. This discovery opens up new avenues for both disease diagnosis and targeted therapies, potentially revolutionizing our approach to treating a wide range of conditions associated with zinc metabolism disruptions. Dietary or naturally obtained zinc has a marginal, if not negligible, effect on suppressing tumor cell proliferation. Metallomix theory on the role of light isotopes in the proper functioning of cells explains that dietary and naturally obtained zinc likely has a mix of light and heavy isotopes. In the case of processed food and other nutrients, the proportion of light to heavy isotopes may further favor heavy isotopes which are associated with disease pathogenesis. In vitro studies of multiple light isotope and amino acid complexes demonstrated the most drastic and long-lasting improvement, measured by a reduction in live leukemia cells, with ZN64 and the aspartic acid complex. Notably, the ZN64 and glutamic acid complex had similarly beneficial properties, but with a less drastic reduction in the number of leukemia cells present. Notably, the naturally obtained zinc complexes, simulating dietary intake, had a nearly negligible effect, which was short-lived and easily counteracted by the cancer pathophysiology. This provides further evidence that demonstrates the importance of purified light isotope zinc complexes as a therapeutic, as dietary zinc cannot provide the same protection due to the presence of pro-tumor heavy isotopes of zinc, which cancer cells can utilize. Our research has yielded intriguing results demonstrating the ability for KLS1 to reduce both local and systemic inflammation, a key factor in many chronic diseases. KLS1 therapy has shown promising metabolic effects, including lowered cholesterol, triglycerides, and free fatty acids, as well as resolving fat deposits in the liver. Importantly, it has normalized the morphofunctional properties of the pancreas and liver, while also regulating the secretion of beta cells in the pancreas and insulin levels. The treatment with KLS1 has shown additional benefits by inducing testosterone and serotonin production and lowering body mass index. In the realm of neurology, Metallomix approach has increased Th-positive dopaminergic neurons, reduced neural damage, and improved brain recovery, leading to enhanced cognitive and motor functions. Furthermore, KLS1 has demonstrated the ability to induce wild-type p53 and PHA-induced T-cell proliferation, suggesting potential applications in oncology and immunology. These wide-ranging effects underscore the potential of Metallomix isotope selective modulation platform to address multiple aspects of health and disease simultaneously. Further evidence supporting the role of light versus heavy isotopes in cancer cell tumorigenesis. Relative to the untreated control cells, the heavy isotope zinc-70 treated cells greatly accelerate the growth and development of tumor cells, whereas the light isotope zinc-64 treated cells markedly decrease the number of tumor cells present. This is direct evidence supporting the mechanism of action of KLS1, which leverages light isotopes of zinc to combat malignant tissues. Additionally, it demonstrates that natural or dietary zinc may worsen outcomes in oncology indications. In oncology, many biological pathways may contribute to pro- or anti-tumor effects. The role of light or heavy isotopes of critical cell nutrients is upstream of most of these pathways. Therefore, the therapeutic effect of light isotopes of zinc may act upstream of many current anti-cancer therapies. Following our in vitro studies, we conducted in vivo studies in rodents which confirmed our previous findings. Purified light isotope zinc-64 and not naturally occurring zinc reduced tumor size and the number of living cancer cells in the animal model. The L1210 cell line used here was injected into the rodents alongside different doses of the light isotope zinc-64 aspartate complex. Interestingly, in this model, there was an inverse dose response whereby the lower doses had the largest effect, whereas the positive control, doxorubicin chemotherapy agent, had little to no effect at all in the time frame under observation. Whether this dose-response relationship is translatable to humans is something we will investigate. In another in vivo rodent model, we noticed a complete remission of melanoma in mice 3 weeks after a single KLS1 injection, indicating a disease-modifying effect. Additionally, given that this investigation was in yet another form of cancer, the evidence supports the role of light isotope modulation as a general anti-cancer therapeutic due to its highly upstream mechanism of action. As a light isotope modulation therapy, KLS1 acts at the key junctures of cellular physiology, gene expression, and protein translation itself. Further investigation into the melanoma tumor morphology of mice treated with and without KLS1 demonstrates a marked reduction in clearly cancerous cells as early as 45 minutes after injection. This effect is maintained over 24 hours. This may suggest early anti-tumor effects driven by improvements in the immune system's ability to recognize and target cancerous cells, or simply apoptosis of the cancer cells. One plausible explanation is that there is a near-immediate effect of light isotope modulation in cancer cells due to the re-engagement of pro-apoptotic proteins and signals. In a mouse-specific cancer model, KLS1 is able to extend the lifespans of mice. This is important because although Ehrlich carcinoma is a mouse cancer, it can form both solid or liquid tumors. This is also a non-differentiated tumor line with rapid proliferation, intended to simulate aggressive cancers in humans. Notably, 100% of mice survived well past the survival time of the control mice. Altogether, these results demonstrate converging evidence supporting a general anti-tumor effect of KLS1 that will likely translate well to humans with diverse oncological indications. Cancers and neurodegenerative diseases are, in many ways, two sides of the same coin. These disorders both involve aberrant gene expression and protein translation. For cancers, this leads to cells that refuse to die when they are supposed to keep the tissue and organism healthy. Whereas in neurodegenerative diseases, this leads to cells that die far too early, and not when they are supposed to. For example, neurons. The difference between regular cells and neurons is a difference in chromatin architecture and therefore gene expression. Regular cells, which replace themselves from adult stem cell populations, are meant to be destroyed when they become cancerous. Neurons have a gene expression program that is meant to ensure they survive for the entirety of the organism's lifespan, because they cannot be replaced by one's natural adult stem cell populations. Therefore, therapeutics that act upstream of both cancer and neurodegenerative pathophysiology likely work on similar cellular pathways involved in the regulation of gene expression and protein translation. In an LPS-induced rodent model of Parkinson's disease, the second most common neurodegenerative disease, KLS1 is clearly able to protect neurons from the ongoing effects of a neuroinflammation meant to recreate the neurodegenerative cascade of Parkinson's disease. The ability to protect neurons from neurotoxic effects is critical to treating and preventing Parkinson's disease by stopping the neuron death as soon as possible. This is a good model of Parkinson's disease because neuroinflammatory effects likely underlie the early and late causes of neurodegeneration in Parkinson's disease, based on human studies of neuroinflammation in patients with Parkinson's disease. Not only did KLS1 protect dopaminergic neurons from the neurodegenerative cascade in the 6-OHDA rodent model, the results demonstrated that animals had more neurons than expected after inducing Parkinson's-like neuroinflammation in the animals. Specifically, the animals that were not given any treatment saw a drastic reduction in the number of dopaminergic neurons. Therapeutics that are only neuroprotective would show a value below the intact animals and above the LPS-only group, untreated rodents. In our study, KLS1 was able to apparently restore the number of viable dopaminergic neurons slightly beyond that of the intact animals. This suggests that light isotope modulation therapy may induce neurogenesis, at least in rodents. We will investigate this in human cell models derived from patients with neurodegenerative disease and potentially in clinical studies too. Supporting the neuroprotective and potential neurogenesis effects of KLS1 as measured by cell counts in the previous slide, we demonstrated an improvement in motor behavior in a PD rodent model. When mice lose dopaminergic neurons, their motor function is not only impaired but also tends to exhibit increased rotational rates, as if the mice cannot fully control their motor system anymore. Treatment with KLS1 drastically reduced the rotation rate of the LPS-induced PD mice, further supporting the neuron count findings that demonstrate neuronal survival and potentially an increase in dopaminergic neurons themselves. This converging evidence supports the role of light isotopes in helping protect our neuronal tissues and even heal them after injury. The LPS-induced PD model is notable because neuroinflammation is a part of neurodegeneration in many dementias, motor disorders, and other CNS pathologies, including stroke, traumatic head injury, and even neuropsychiatric indications. Importantly, KLS1 also reduced neuroinflammation. This is especially important in an LPS-induced model of PD. We can see clearly that KLS1 reduced GFAP, a protein that is released into the blood when astrocytes are activated due to neuronal damage or death. The reduction in both peripheral blood and CNS GFAP levels indicates that KLS1 is preventing and ameliorating neuronal damage and death, preventing the activation of astrocytes and therefore reducing the amount of GFAP released. This reduction in neuroinflammation is more than just a direct effect on inflammatory molecules. It is a consequence of therapeutic activity upstream of neurodegenerative pathways. This demonstrates that KLS1 does not impair or suppress the immune system, but rather stops or controls neurodegenerative damage that would activate an immune response via astrocyte activation and the recruitment of microglia in the CNS. Although we did not measure glial recruitment in the LPS-induced PD rodent model, for example, via measuring YKL40, we observed in a rodent model of Alzheimer's disease, AD, that KLS1 can decrease the activity and recruitment of monocytes after inducing AD-like symptoms with toxic beta-amyloid oligomers. The reduction in phagocytic activity likely reflects a reduction in neurodegenerative pathophysiology, which would otherwise activate many monocytes, such as microglia in the brain. This is an important finding because immune cell activation in neurodegenerative diseases is a key part of the disease cascade and a main contributor of neuronal stress and death. KLS1 is able to reduce phagocytic activity without suppressing the immune system, thereby avoiding potential side effects associated with immunosuppression. In the AD rodent model, we also observe behavioral and cognitive improvements after treatment with KLS1. This further supports KLS1's upstream therapeutic effect in neurodegenerative diseases. In this case, we used the Barnes-Maze test, which is a well-established paradigm to test behavior, spatial learning, and cognition in rodents. In this model, amyloid beta impairs the rodent's ability to find objects in space, which is a function of their spatial cognition and memory. Treatment with KLS1 is able to largely reverse the effects of toxic amyloid on neurons. Cancers, neurodegenerative diseases, and metabolic diseases all share alterations in metabolism in their disease pathogenesis. This is observed in both animal models and human patients with these diseases. Therefore, a disease-modifying therapy should address disease-related metabolic alterations. In another rodent model of metabolic diseases, we observed that obese animals treated with KLS1 were able to reduce their weight without impacting their calorie consumption and appetite. Metabolic disorders and obesity are strongly associated with metabolic impairments. In other words, the body is unable to normally metabolize macromolecules and distribute them throughout the body to tissues that need it. Treatment with KLS1 appears to correct this issue without superficially targeting the signaling pathways for satiety, which many currently available drugs for metabolic disorders and obesity do. Further converging evidence on the beneficial effects of KLS1 in metabolic disorders can be observed here. We see that inducing obesity with a high-calorie diet causes the largest weight gain, as expected, whereas treatment with KLS1 and a high-calorie diet clearly and markedly reduces the total weight gain over time, despite the continuation of the high-calorie diet. Importantly, we also observe that when KLS1 is given to control animals, no high-calorie diet, they do not lose weight. Therefore, KLS1 corrects metabolic function without directly forcing cells and tissue to shed weight, like a stimulant-based approach to weight loss would cause. This is an important finding because it suggests KLS1 normalizes metabolism upstream of the targets of therapies that target the sensation of satiety. This would eliminate or reduce potential side effects that are common to weight loss drugs today. The metabolic improvements driven by treatment with KLS1 are further reflected in the normalization of several laboratory results related to liver function, which are key safety indicators in clinical trials. These results provide an objective rationale for the safety of KLS1 in not only metabolic disorders but also cancers and neurodegenerative diseases. These results highlight KLS1's beneficial effects on lipid metabolism, which is notable because lipid metabolism is impaired in cancers, neurodegenerative diseases, and metabolic disorders. Furthermore, The beneficial effects of KLS1 on metabolic function are reflected in normalized insulin sensitivity and glucose levels. While this benefit may be obvious in metabolic disorders, notably without needing to resort to therapies that directly target insulin or metabolism-regulating hormones, there is also a connection to cancers and neurodegenerative disorders. In both, aberrant glucose metabolism underlies several parts of disease pathophysiology in cancers, Alzheimer's disease, and Parkinson's disease. With a strong intellectual property portfolio, Metallomix is well positioned to capture a substantial market share. Our patents cover the composition of matter and methods of use, providing a first-mover advantage in cold isotope therapeutics. We have 5 issued patents, 8 indication-specific patent applications, and over 16 provisional patent applications. Our issued patents are valid until 2035-2039. Metallomix leverages advanced machine learning and AI technologies to optimize clinical trials and enhance target discovery. Through our collaboration with Numinos and its Cure platform, we utilize a comprehensive clinicogenomic foundation model with 100 million parameters to identify causal targets and predict treatment effects. By integrating public and proprietary datasets, we significantly improve biomarker discovery and clinical trial outcomes. This technology-driven approach ensures efficient drug development through intelligent patient selection and optimized clinical trial design. By predicting efficacious primary and secondary endpoints, we substantially increase the likelihood of positive trial outcomes. Our AI-powered strategy enables both reverse and forward translation, allowing us to predict untaken treatment effects even within limited sample sizes and high-dimensional data. We are currently raising $130 million in Series A funding to advance our platform and pipeline. This investment will support key milestones and drive our mission to revolutionize disease treatment through Metallomix. By securing the necessary resources, we can continue to innovate and expand our therapeutic offerings, ultimately improving patient outcomes. Metallomix is poised for significant clinical progress in the coming years, with a robust pipeline spanning multiple therapeutic areas. Our clinical development plan includes the first patient in for Phase 1 oncology clinical trials initiated in Q3 2024, marking a crucial milestone in our program. Following this, we anticipate commencing Phase 1 clinical trials for neurology indications in Q3 2025, closely followed by the initiation of Phase 1 trials for metabolic indications in Q4 2025. The company expects to report top-line data from their Phase 1 oncology trials by Q4 2025, providing valuable insights into the safety and preliminary efficacy of their innovative therapies. Concurrently, we are conducting ongoing mechanism of action studies to support our clinical indications, ensuring a strong scientific foundation for our therapeutic approach. Additionally, we are engaged in continuous studies aimed at expanding our clinical pipeline, demonstrating our commitment to addressing a wide range of unmet medical needs through the isotope selective modulation platform. Metallomix's diverse pipeline targets a substantial and rapidly growing market across multiple therapeutic areas. Our current pipeline addresses a global market projected to reach an impressive $211 billion by 2030, with an average compound annual growth rate of 8%. This market is segmented into 3 key areas. Neurology pipeline, the largest segment, with an estimated market size of $121.9 billion by 2030, growing at a robust average compound annual growth rate of 9.6%. Oncology pipeline, projected to reach $31.0 billion by 2030, with an average compound annual growth rate of 6.3%. Metabolic pipeline, expected to grow to $58.5 billion by 2030, with an average compound annual growth rate of 7.8%. These projections underscore the significant market opportunity for Metallomix's innovative isotope selective modulation platform across multiple high-value therapeutic areas, positioning the company for substantial growth in the coming years. Metallomix is pioneering a revolutionary approach to drug development with its new class of isotope selective modulation therapies. Our innovative platform leverages isotope effects as a therapeutic mechanism, offering a fully defined, multi-pronged mode of action based on the broad therapeutic potential of zinc. The company's lead candidate, KLS1, exemplifies this approach and is being developed through a straightforward and efficient path to commercialization. This strategy can be replicated for future drug candidates, creating a rinse and repeat model for expanding the pipeline. Metallomix's unique understanding of disease etiology and pathogenesis underpins this approach, supported by issued and pending composition of matter and methods of use patents in the U.S. and internationally. The platform's rigorous scalability allows for expansion into new indications and therapeutic areas through the development of new ISM formulations. Metallomix is pursuing the 505(b)(2) regulatory pathway for KLS1, highlighting its novel strength in modulating rates of cellular uptake and enzymatic function. The company is also seeking fast-track designation for combination therapies in oncology and considering breakthrough therapy designation for KLS1 in chronic lymphocytic leukemia, underscoring the potential of this innovative approach to address significant unmet medical needs. At Metallomix, we assembled a formidable leadership team with diverse expertise to drive its innovative approach in Metallomix-based therapeutics. Dr. Max Temnick, the founder, president, and CSO, brings a wealth of experience as an investor in biotech startups and a seasoned entrepreneur with a strong background in chemistry. Dr. Sergei Petokov, serving as co-founder and CEO, contributes his distinguished track record as a venture capitalist in the biotech sector, known for securing unicorn IPO exits and mergers and acquisitions. The company's medical expertise is anchored by co-founder and CMO Dr. Santosh Kasari, a leading neuro-oncologist in the U.S. with extensive research and development experience. Rounding out the executive team is co-founder and CFO Sergey Gurin, MBA, an accomplished serial entrepreneur and investor with expertise in management, business growth, intellectual properties, and securities offerings. This team of seasoned, mission-focused specialists consistently explores and assesses a broad range of resources to support Metallomix's business expansion and strengthen its operations, positioning the company for success in developing breakthrough therapies. Thank you for your interest in Metallomix. We are excited about the future of Metallomix and our role in transforming therapeutic approaches to complex diseases. As we look to the future, we are excited about the potential of Metallomix to transform disease treatment. Our innovative therapies offer new hope for patients and represent a significant step forward in healthcare. We are committed to